I'm trained as a scientist, and so of course today what I want to share with you is some science that you can do at home because science is all around us. I'm originally from Puerto Rico, and growing so what I hope to do today is actually share some activities that you can do at home with things that you can find in your fridge and in your pantry, and so that you can, fall in love with science as well.
Very cool. Excellent. So let's just go ahead and have you get started, and then I'll have some follow-up questions for you more about the work that you do and the importance of science writing and education too. So let's have you go ahead and get started. I'll throw some questions at you as we go.
Sounds good. So today what we're going to do is we're going to extract genetic material or DNA from strawberries. As you know, DNA is where all the instructions for every living organism are. It's basically our coding of instructions. We have DNA in each one of our cells. Now, I can't really extract DNA from myself or any other animal right now, so what we're going to do is we're going to use strawberries. So I have some strawberries here, and what we're going to do is use some salt, which I've put over here, just water, some dish soaps, or they can be glass if you want to be really green. And then we're just going to get the strawberry DNA out of the fruit. So what you start is you take the fruit and you take all of the leaves off. Strawberries have 8 copies of a genome. So the genome is of the genes that each person or each organism has. And so strawberries have 8 copies of them. We have 2. So strawberries are really great to extract DNA because there's just a lot of DNA in each one of their cells. So what you're going to do is take the leaves off or any of the green stuff on the top, because although the green leaves, the leaves at the top, they have DNA, their cells are tougher to break, and so it's harder to get the DNA out of them. So what we're going to do is take a Ziploc bag, put a few strawberries in there. Let's see, I'll put 4. The more strawberries you put in there, the more DNA that you'll get out. And what we'll do first is seal the bag, and then we're just going to smash the fruit. You can do it with your hands. I'm not going to go and pound on them because I don't want to be too noisy, but you want to really break the fruit down. Starting to break, let me get some of the air out of this so it's easier. So by breaking them down, what you're doing is actually getting some of their cells open and starting to get some of DNA so that it becomes accessible. Ooh. I don't want to break the bag, let's see. Yeah, so when you're doing this at home, try to get as much as out of the bag as possible, so that it's easier to do this.
And is this an activity that you lead with, like with youth, try to get them interested in science? Like what are you going to use this for normally?
Yeah, you can definitely use this to demonstrate to kids and adults alike how DNA extracting technology works. So what we're doing here is we're using really simple chemicals that you can find and reagents that you can find at home to do this, but the actual process that I'm doing right now is not very different from what scientists do when they're extracting DNA from organisms in the lab. The principles that make this work are not very different from what scientists do in the lab. And in fact, if you think about the human genome, for example, something similar to this process is what scientists did to get DNA out of samples from different people to then get their genetic information and sequence their genome. So this is a really cool, fun, hands-on way to show people how you get DNA out of cells in an organism and then afterwards you can take that DNA and actually sequence it and get more information about the genetics of any organism.
Very cool.
All right, so I think it's all smushed in it. It really looks like a smoothie in a bag. What I'm going to do is, in this plastic cup, I've put half a cup of water. What I'm going to do with that, I'm going to put the salt in it. So there's a teaspoon of salt, and I'm going to put it in there. And then I have a teaspoon marshmallow and any kind that you have at home, you can just use that to do this experiment. So I'm just mixing it up. I'm just making a solution. This is going to be my DNA extracting solution.
Sorry, there's a little bit of a delay on my end, Monica. So what is the, could you repeat, maybe I missed it, what the dishwashing soap does in this process?
So what the dishwashing, so the walls of your cell, so the DNA is inside your cell, and within each cell there's a nucleus, and that's where the DNA lives. And the walls of the nucleus and of your cell are basically made out of fatty molecules. And what the dish soap, just like the dish soap gets the grease off of your plates or your cups or your dishes, it's going to help break up the cell wall so that the DNA can come out. So basically it just tears apart all the fatty molecules that are on the... So next up, we are going to take, let's see, I don't want to spill that all over my desk. Can you hear me okay?
Yeah, there's a little bit of a lag, so I'm just going to let you talk, and then I'll try to catch up a little bit.
Okay, sounds good. That's good. All right, so I have a good mix over here, and then I'm going to take that same teaspoon and put 2 teaspoons of my DNA extracting mix into the bag. So there's 1 and 2.
And can you hold that up a little bit higher so we can see that on camera?
A DNA extracting mix, water, table salt, and dishwashing liquid. And now I've put 2 teaspoons of that mix in here in the bag where all the smushed strawberries are. So I'm going to close that again and then smash this. So I'm basically a dish at the so in there and do their job and break the cells further. Let's see. Look at that. So now it's a soapy strawberry smoothie.
Not for consumption.
No, not for consumption. I mean, you could try, but it's not going to be fun. All right, so I think that should be good. The other materials that we need is a coffee filter. So just any coffee filter that you have at home. This is Seattle, so people do drink a lot of coffee, so you'll likely will have a coffee filter at home. So what I've done is put the coffee filter at the top of a plastic cup. And, I've had to be resourceful and put some of these pins here to hold it because I don't have an assistant, unfortunately. And so what the coffee filter is going to do is going to filter out the bigger chunks of the strawberries that were not mashed up, the strawberry cells. So let's do that. This is the messy part. Alrighty. Let's see. Wow. Alright. And I'll show you in a minute or in a second how that looks. Alright. So I just poured all of the strawberry or most of the strawberry dishwashing liquid smoothie in here. And what I'm going to do, oh no! See, this happens all the time with scientists. Accidents happen. That's why you have all that protective equipment so that you don't get hurt.
This was
Uh,
most of my high school lab experiments, is me messing it up along the way.
yeah, that's okay. Scientists mess up a lot as part of it. All right, so what I'm doing now is I'm taking the filter and I'm just squishing it so that I can get the liquid.
See, I think we might have lost Monica there, so we'll try to get her back. Oh wait, there she. Are you back on, Monica? Oh,
Yeah,
sorry, it
sorry.
was a delay on mine. If maybe it could have just been on my feed, but would you mind repeating that last step for me, or just
Yeah, so
recapping the
what I'm
last
doing,
step?
and what I'm doing right now, and as I'm actually squeezing and getting all dirty, I'm actually squeezing the liquid through the filter so that we can actually get it into the cup. And I'm going to get rid of some of that because if we have the DNA that my really dirty hands full of strawberry. It smells really good, but I'm sure it doesn't taste very well. So the last part, which should give us the DNA, so there's the filtered strawberry liquid. And what we're going to do now is so before we started, I put some 90% rubbing alcohol in the freezer. And what we're going to do now, and now I've had it on ice on the side, and what we're going to do now is, first of all, open it. And what we're going to do now, what the alcohol is going to do is actually, because DNA doesn't like alcohol, basically, it's just going to clump all together. And by pouring the alcohol into the cup, we should be able to just see the DNA. So hopefully this will work. Let's see. All right. All right. I think we can see it. So it's when you have a lot more, I don't know if you can see it, but it's lighter at the top.
Oh yeah.
And darker at the bottom. Can you see that?
Mhm.
So what's happening is that all of the unbroken cells and the heavy parts of the cell, so each cell has organelles like the mitochondria, which is most of other organelles that produce important molecules in the cell, because they're berries that were not broken down really well. It's all going to the bottom because it's heavier, and the alcohol is actually causing the DNA to clump together at the top, and that's why it's actually clear at the top, and the clumps that are forming. So when it's lighter at the top, so the DNA is kind of floating on after. So I have just a coffee stirrer. What I'm going to do now, oh wow, there's a lot, is pull the DNA out.
Whoa.
So there you have strawberry DNA.
Very cool.
Yeah, so if you were in a lab, actually when I was during my PhD, when I was doing my experiments in the lab, I had to work with DNA a lot, so I had to extract DNA from cells to genotype animals, so to know if my animals were a mutant or were not. And the process that I actually went through to do that was not much different than this. In the old times before, now there's kits and machines that will do this for you in a shorter amount of time, and you don't need as much material. But way, way back, this is pretty much what people would do. They would use alcohol and a mixture of salts and a soap that will break down the cells to get the DNA out. But the cool thing about this experiment is that you can actually see the DNA.
Very cool. And how does this different than when people do their own genealogical DNA testing or human DNA testing? Is it a similar process, or does it look different for human DNA?
From your dog or your cat is slightly different from what you have in terms of the genes that you have, but the building blocks of the DNA, it's the same for all organisms, from bacteria to humans. So although the composition of the genes and the coding of the genes is slightly different, the actual building blocks, which are the nucleotides, are very similar, and also the structure of the DNA is very, very similar across organisms. So the process to actually extract DNA from bacteria all the way to humans is very similar. You know, you have to use salts and soap, something to break down the cells, and then something to actually get the DNA out of all that mixture, and that's pretty much the same for all organisms. So what you would do if you now have the DNA, and this is very, because this is kind of a down and dirty process. So you contaminate pure agarose gel, and so because DNA is negatively charged, you can actually run the DNA on that gel, and so that you can see it. And then if the DNA, different genes or different parts of different genes have different sizes, and depending on the size of the chunk of DNA, then scientists can determine what gene is this, what part of the gene this is, and then you can do other kind of procedures to get more into the details of the structure of the DNA and so on.
Very cool. And I know that science writing and science journalism is a really important part of your work, and you've done amazing articles on the Globalist I'd encourage people to check out. Can you tell me more about why you're passionate about journalism and storytelling and science as it relates to the Seattle Globalist?
Yeah, so things that I really appreciate about the Seattle Globalist is the support, not just with our peers, with the general public, with the people that are actually from the discoveries that we're making. And so spaces like the Globalist allow people like myself who are outreach scientists and communicators and writers and journalists to bring the science to people, to share that knowledge, to let them know how science is all around them, how science is actually happening in their community. The UW it's our neighbor in Seattle, and there's amazing science happening at the university, and the people in Seattle should know what's happening, should know what we're doing, and should know how is it going to benefit.
Do I still have you, Monica?
Yes.
Oh, sorry, it was a long delay on that one. I apologize.
That's okay.
I think it's
Did
okay.
you hear the last thing I said?
Yeah. Could you repeat that one more time? It kind of stuttered through on the audio part for me. Could you repeat one more time the last part?
Sorry, that's okay. I'm sorry. I hope my connection.
Oh, no worries at all.
What I was saying is that science is a global enterprise, and it goes beyond geographical and physical frontiers, and it's a collaborative effort. And that spirit of collaboration and of globalism is very much in line with what the mission and the values of the Seattle Globalist. So it's a great opportunity to be able to share science with the globalist audience.